in the article titled Use of Gene Editing to Introduce the Pole Trait into Elite Germplasm by Allison Van Ennenem and Macy Mueller from the University of California, Davis. The authors suggest that horned cattle within the dairy industry pose a problem for producers because of the possibility of injury to the cattle and the worker. With this being said, many people view physical dehorning as a painful experience for cattle. A solution to this is CRISPR-Cas9. CRISPR stands for clustered, regularly interspaced short palindromic repeats, and Cas9 stands for CRISPR-associated systems. However, people also question whether this gene modification method is an ethical solution to physical dehorning. So, what do you think about CRISPR being used in the dairy industry?
The polled horn trait in cattle is actually due to a long non-coding RNA, but for simplicity's sake, it will be referred to as a dominant or recessive allele. The horn gene is inherited as an autosomal recessive allele, so this combination of a big P and a little p would result in a polled individual, while this combination of two little p alleles would result in a horned individual.
When brought together, these two items create an RNA-mediated system for gene silencing that is similar to restriction enzyme systems, but are far more adaptive. CRISPR-Cas9 is a naturally occurring phenomenon that provides prokaryotes with adaptive immunity against viruses and plasmids by using CRISPR RNAs, or cRNAs, to guide the silencing of invading nucleic acids. So now that you know what it does, what exactly is it? CRISPR is a short RNA sequence that reads similarly backwards and forwards, hence palindromic in the name. Cas proteins add viral or plasmid sequences, spacer RNA, between CRISPRs as new invading DNA is encountered. Transcribed spacer RNA forms double-stranded RNA or RNA-DNA hybrids. Cas proteins, endonucleases, cut the double-stranded RNA or RNA-DNA hybrids for defense. With this system being so efficient, scientists have been able to synthesize the same pathway to make specific and calculated changes to a genome. Applications for this would be to add or replace genes or parts of genes, such as that with the horned pole gene in dairy cattle. This works by inserting donor RNA into where the cutout RNA used to be.
Unfortunately, as with all new technology, CRISPR-Cas9 has some disadvantages. A disadvantage is that each CRISPR-Cas9 is its own modification, or drug, that must be approved by the FDA, which could take years. Along with the disadvantages, CRISPR-Cas9 brings ethical issues into question. One such question is that unintended changes will be making to genomes, and will those changes be harmful?
Yeah!